Thank you very much, Andrea, for agreeing to give a Build a Cell seminar, and you can get started.
Thank you. Let me share again. All right. So, hello everyone. Good afternoon, good morning, good evening, in case you are a bit further away. And thank you for coming to this presentation where we'll be talking about my recent work on enzymatically synthesized artificial cells. Personally, just to give you a quick overview of what I do, who I am a group leader at the Technische Universität Darmstadt in Germany since last year. I did my PhD in Switzerland, in Basel. Then I moved on to Prague and then Glasgow for my postdoctoral work before joining the University of Darmstadt. So, without further ado, let's talk about a bit more about science. Today, the audience is one of those rare audiences where I don't have to make a preface about what artificial cells are and why we would want to have them. But it's necessary to give a preface for people who have never heard of Darmstadt. Where are we? We are here in Germany, right almost in the middle. This is our campus, very much buried into the woods. You can see the area where my labs and offices are. And what is interesting is that right next from Darmstadt, there is this castle of Frankenstein that I always found fitting in our quest for making hybrid or artificial cells, or giving life to what isn't alive. And when talking about artificial cells, there are many ways we can look at it, many perspectives. My main interest is the container. Why am I interested in the structure, in something that is actually making the compartment of an artificial cell? If you look at one of the most famous theories about the development of life, the so-called guard model, it posits that you need some specific molecules that can harbor compositional information and allow catalysis and thus the evolution of molecules that are not biotic yet. This has led to the plethora of artificial cell work that has been developed in the past decade, where you had many aspects of cell have been mimicked and then even added upon. And also can look at the top-down approach, where actual real cells have been made simpler and simpler up to the bare minimum synthetic cells that can still act as much. However, the great majority of these artificial cells have share the same biochemistry in their container, in the actual structural molecules of the real cells. It is the lipids and phospholipids that form the membranes. So these are vesicles that are made of the same or very similar lipids to what we found in nature. An interesting twist upon this is looking at polymers instead of lipids. So polymers that are amphiphilic as well as much as the lipids are, and thus self-assemble into structures that will have a hydrophilic core, hydrophobic membrane, and then, of course, an aqueous environment outside. Much like liposomes, these polymer zones can encapsulate both hydrophilic and hydrophobic molecules. They have been developed to be biocompatible in case you want to have biomedical applications. But compared to classical liposomes, they tend to offer a higher stability, both physical and chemical side, and also they have a nicer versatility when you want to modify the membranes. This is not new. Artificial cells have been made out of polymer membranes, have already been developed several years ago. You can see a paper from 12 years ago where they could express a fluorescent protein inside these artificial cells made out of amphiphilic block copolymers. However, all these polymers are synthesized in classical ways. And by classical ways, I mean there must be a chemist that synthesizes the polymer in blocks, and then they have to be assembled somehow, either by using microfluidics or any kind of other system to force the assembly of these vesicles and encapsulation of biological components, for instance. However, there is a way where to form vesicles and 3D nanostructures that is much closer to how lipid structures are formed in nature. That is the polymerization-induced self-assembly, where you start with a solvophilic polymer, that is a polymer that is soluble in your environment, which is usually aqueous, so water, on which you extend a core-forming monomer, so a monomer that is hydrophilic as a monomer, but as the chain is expanded and this block of new monomer gets longer and longer, it becomes amphiphilic. So you have a hydrophilic block and a hydrophobic block. This amphiphilicity leads to the self-assembly of this molecule as it gets formed, as the polymer is actually synthesized into structures that go from classical spheres, or even just micelles, to tubular micelles or worm-like micelles, up to vesicles that actually have an aqueous core. You can see here a paper from a few years back. We're using photocatalysis. You can actually see these vesicles growing as you shine light in the area where the polymers and the initiators are present. However, again, this is mostly made using classical organic catalysis. There is, however, a concept in controlled radical polymerizations, which are the kind of polymerization that are water tolerant, that are used to make these vesicles, which is to use some proteins and some enzymes specifically that can do these polymerizations instead of using classical metallo-organic compounds, for example. Using some metalloproteins, such as even hemoglobin or horseradish peroxidase, lacase, and whatnot, you can actually perform this kind of radical polymerization called ATRP that forms using an initiator that is usually containing an halide, and in aqueous environment with ascorbic acid or similar reducing agent, you get amphiphilic block copolymer or a block copolymer or a copolymer in general. Then this is what actually made me come to first Glasgow and then Darmstadt, which is the specialty of the group of Professor Nico Bruns. Thus, we decided, together with my collaborator in Freiburg, in Switzerland, Dr. Jimaja, to sort of translate this concept of enzymatic polymerization for classical soluble polymers to produce amphiphilic block copolymers that would ideally be extended as they would be synthesized. As catalyst, in this case, we used hemoglobin. So hemoglobin is not classically seen as an enzyme, but it does have a peroxidase activity, which is what you usually need in order to extend the polymer from bromine end, for example. And then we were trying to achieve these diblock copolymer nanostructures. Chemically speaking, talking about molecules, we used a classical PEG-based microinitiator having bromide, and as monomer, hydroxypropyl methacrylate, one of those monomers that are water-soluble as monomers, but then become hydrophobic as they become polymerized. So using myoglobin, sodium ascorbate as the reducing agent, and a solvent made by 95% of buffer PBS, plus 5% of a co-solvent DMSO or DMF, working both, we would achieve this block copolymer. And by varying both the amount of monomer in the solution and the ratio between the macroinitiator and the monomer, that is the aim degree of polymerization, we explored the so-called pizza space, looking at the various morphologies that were formed. At very low content, would get spheres, nanoparticles. Increasing the amounts of monomer, we would get worm-like mice. We had quite of a broad mixed phase, but very interestingly, we could get vesicles. We could get vesicles at high contents and high ratios of monomer to initiator, thus meaning that this enzymatic reaction could form vesicles. Now, if you look at this, you can see in this area, we got vesicles with a membrane that was quite thick, 30 to 50 nanometers, so quite much thicker than normal liposomes, but still was permeable to up to roughly 1000 Daltons. You can see the result of a reaction is a very thick and dense reaction with an estimated amount of vesicles 10 to the 8th vesicles per ml. So pretty nice and pretty high yield. This is how they look like at the confocal microscope. The red is coming from the labeling of the dye using a cholesterol PEG dye that inserts into the hydrophobic membrane. They are pretty diverse in shape and size. Many of them are quite elongated, so much that some of them are also so small that they could be visualized in TEM, like I showed before. Others are better to be visualized via laser microscopy, but still they do look like vesicles, and they can encapsulate macromolecules. Smaller molecules will tend to go through, for example, fluorescein goes in and out very easily, but bigger molecules like this labeled dextrane can be added to the reaction mixture. It would be encapsulated, and then you just need to purify the vesicles either by centrifugation, size exclusion chromatography, dialysis, whatnot, in order to retain the macromolecules inside. This encapsulation is, say, the encapsulation efficiency dependent on the size of the species you want to encapsulate. Of course, myoglobin, the catalyst, will self-encapsulate because it's already present into the solution with a nice 80% encapsulation efficiency. Bigger molecules will be slightly less efficient, but still encapsulated. And you can even get nanoparticles that are encapsulated in your vesicles, like these Alexa 4 or 5 silica nanoparticles. If you look at the shape and size, and then this I did by plotting the area of these vesicles, so compared to the aspect ratio, where the aspect ratio in this case was the minimum ferrate diameter divided by the maximum ferrate diameter. Did that because most of them were not really very circular, so it made more sense to look at the total area in square micrometers. You see that if you take the ones having nothing else, only myoglobin, shape and size are a bit all over the place. Bigger ones tend to be slightly more elongated, that is at lower aspect ratios, but still a bit all over the place. Same thing for macromolecules like FITC dextran or beta-galactosidase. If you look instead at the cargo of nanoparticles, there is some kind of interaction between the polymer and the nanoparticle that sort of makes the vesicles slightly rounder and on average slightly smaller compared to others. Still, it's still not a fully monodisperse sample. If you look at the micrometer encapsulation of micrometer objects, you can encapsulate polystyrene microparticles, roughly 2 micrometers in diameter. You can see that you do have encapsulation, even though now the interactions during the self-assembly sort of influence also the shape of these vesicles. And even more so, you can encapsulate bacteria. In this case, use the fluorescent bacteria expressing enclover. Again, here you get really wild shapes and sizes, but still you do get fluorescent bacteria within your vesicles. If you look at the viability of your bacteria, this was done using fluorescent diacetate and propylidomide staining, or non-fluorescent bacteria, of course. You do have, of course, some dead bacteria, because there is some interaction between the growing polymer chains and bacteria, but still you way above the 60 to 70% viable cells after a round of polymerization. Moreover, of course, the enzymes inside are there, they are still active. So if you co-encapsulate, for example, the classical glucose oxidase together with myoglobin, which has peroxidase activity, and then you do the very classical cascade reaction with glucose being metabolized by the oxidase that produces hydrogen peroxide. This is then used by myoglobin to produce resorphin from ampersand. You can see here in this time series how the fluorescence is initially localized within the vesicles, and then it diffuses out. Beyond that, just microreactors would have been easy, but would have been very simple. What was more interesting for us was to develop these artificial cells to something that was actually resembling what cells do. And the classical and the obvious thing is to look at the production of proteins. So the expression of proteins using a non-living system. In this case, we encapsulated cell lysate from E. coli with a plasmid that was encoding for several proteins, in this case for the fluorescent enclover, by feeding amino acid, nucleotides, and also having an energy regeneration system to help drive this reaction forward. We could find fluorescence, green fluorescence in these vesicles. Intriguingly, if we compare the activity of these vesicles immediately as producing protein as soon as you assemble them, and then the same vesicles kept in the fridge for 1 year and then measuring again, we still see that we do get some lowered but still present protein production activity, meaning that these polymers, which is actually known already from the literature, are capable of stabilizing the very sensitive biological components that we are encapsulating. And beyond that, I also tested other proteins, for example, alkaline phosphatase. Now, alkaline phosphatase is a classical protein that is used in colorimetric assays, but it can transform this substrate, which is calcium glycophosphate, into calcium phosphate. This is a non-soluble mineral that then precipitates, thus making this kind of triggered biomineralization. And you can actually see the absorption, this mineral by the absorption of some dyes, such as fluorescein, that makes these very deformed, elongated vesicles, plus some of them which are much smaller and much more irregular shape. You can also see the actual mineral here in this in the top Eppendorf tube that actually precipitates, whereas the same vesicles without the production of protein still have no precipitate inside. Finally, also, we tried to express actin as a cytoskeleton component, so trying to form a structural protein inside the cell. We co-encapsulated it with labeled phalloidin in order to be able to visualize the protein afterwards. And this was quite puzzling because we didn't get the filament we were expecting or some kind of networks, but we got this kind of gel phases maybe, or anyway, interacting in a weird way with the membrane, even forming internal compartments inside, pinching off the membrane from the cell. However, we didn't investigate as much as we would have liked this behavior, but still we could see the production of actin and its polymerization using an osmotic shock with magnesium inside. Now, this was what we did 1 year ago, so published, but then, of course, there are more questions that are arising from all these results. Anytime you do something, you get new venues to explore. One of them is how do we control the morphology of these artificial cells? So something simpler, not looking at what they can do inside proteins or kind of metabolism. It's mostly about the polymer membrane. As we said, there is a high dispersity of these artificial cells. They have bigger, smaller, elongated, rounder, and whatnot. So you can see here the standard bio-pizza reaction. But there are, in these kind of reactions, possibilities to modify, to slow down, in some cases, the polymerization or induce a better dispersity of the polymer chains so that some of them are not longer, some are shorter, but make it a bit closer to a homogeneous dispersity. One of them, for example, is to add a chain transfer agent, a thiolated compound that slows down and allows a slower assembly of these polymer chains. Now, this has been developed for classical ATRP reactions, tried to apply it to this system. And what do we see? We see that the vesicles tend to be much rounder and bigger. So we enrich the big and rounder fraction of our vesicles. On the other hand, if we try to add a bit of hydrogen peroxide in our reaction, and this was quite serendipitous because we were attempting to develop a way to degas our solution because the this polymerization is very oxygen sensitive. We tried to use glucose oxidase as a way to remove oxygen and allow us to work with an open vessel. However, this hydrogen peroxide produced does react with the polymer chain, the growing polymer chains, and so we do get a mixed control radical polymerization, that is ATP, and free radical polymerization coming from the hydrogen peroxide. Now, the polymer chains here tend to be shorter, and the self-assembly stops earlier. You do have these smaller and slightly rounder vesicles because they don't have the time to grow as more elongated ones. So you do shift all the equilibrium towards slightly rounder but much smaller vesicles compared to having a slower polymerization using a chain transfer agent. This allows us to think about making multi-compartments that rely on having bigger and vesicles that harbor smaller ones. As a simple experiment, we made the first bio-pizza making the smaller vesicles, so those that have hydrogen peroxide, with a fluorescent polymer encapsulated into them. Then we took this and added them to the reaction that would lead to bigger vesicles. And we got this. Now, the control of the shape was not as good as it was in a pure reaction with CTA, but we did find fluorescent round objects, which most likely were the vesicles. It was impossible to label specifically that membrane, the membrane of internal vesicle, and not the one with of outside vesicles. But still, you can see internal compartments inside these artificial cells. And just for your curiosity, you can see the distribution of encapsulated vesicles within the bigger ones. Now, beyond that, of course, we made this polymerization enzymatically. And the obvious question I always get when I present my work is, but does it mean that you can do several rounds of polymerization with the catalyst encapsulated inside? Now, this is not as easy as it would look like for several reasons. One of them is that we use a macroinitiator that is bigger than the molecular weight cutoff for this semi-permeable membrane. Secondly, typically these polymer chains are made by living radical polymerizations. They are called living because then you can continue reacting after several steps, more and more polymer on this chain with the bromide. However, what we found by analyzing them is that the polymers still in solution are living chain in the sense that you can extend them. But once they get into, they assemble into the membrane, they are dead. That is, you can't produce them. You can't extend the polymer anymore. So, based on this, I tried to produce a first hydrophilic block that would, again, enzymatically, so using this PEG methacrylate moiety and with this clickable initiator that would make this soluble polymer inside that would not be able to leave because of the size. Then by extending with HPMA and conjugating this the BCO dye on the azide end of the hydrophilic block, we would have this amphiphilic block of polymer inside. Then hopefully, by some reason, we would expect either internal compartments to form because the polymer wouldn't be able to enter the preformed membrane, or form the membrane and then eventually destabilize it and then lead to some kind of enlargement or even budding and something similar. You can see here a vesicle, how it looks like before the polymerization. This is the original membrane. If you stop the polymerization at first step and then you just label the pegma, you can see the polypegma inside the vesicle. If you add hydroxypropyl methacrylate, you can find this new polymer, the green one, the one that is labeled with this green fluorescent dye, on the membrane. You see the vesicles are now either two layers of membrane or one layer with two different polymers. It's not possible to tell yet with this preliminary data. But still, we can produce new membrane on the old vesicles. However, to this point, still I can't find any apparent change either in number or size of these vesicles. So there is still a lot to be investigated. Now for something completely different, the concept of enzymatic polymerization to produce artificial cell or cell-like structures, but not vesicles. This has been made in collaboration with the University of Mainz, close here in Germany, where we produce a cationic polymer, this polydimer, again, enzymatically. Thanks to this cationic character of the polymer, this will complexate ATP, so a phosphate, a polyphosphate, and this will lead to the complex conservation into these phase-separated artificial cells. You can see here how they look like if you take the polymer and mix it together with ATP and fluorescent TAMRA, so BSA with TAMRA labeling. And this polymer can, for example, take up several enzymes. You can see again that BSA as a sort of basic staining, but then you can also get creatine kinase and hexokinase all in the same droplet. Why did we choose these two enzymes here? Because these two enzymes are involved into a cycle where creatine kinase using phosphocreatine can produce ATP, but hexokinase using glucose takes this ATP and produces ADP. And one gives conservation, ADP doesn't. Or better, does to a lesser extent. If you look at the turbidity measured in absorbance at 600 nanometers, you add these two enzymes with phosphocreatine and ADP, you see that creatine kinase is faster and produces ATP at the beginning, leading to the formation of coacervate, but eventually this falls down to the dissolution of this coacervate, because hexokinase takes this phosphate from the ATP for its reaction, and then ADP is not able to coacervate as well. It does to an extent this polymer. And last but not least is this last proof of concept, the enzymatic polymerization used for coacervation, what we call biopic, where we use the same concept as before, so the monomer plus ATP together with our catalyst, in this case again was horseradish peroxidase, not myoglobin, and this together automatically produces coacervate. You can see here the turbidity of our solution after a couple of hours of reaction. And what is very fascinating is that these coacervates are so tightly packed, and that an addition of a fluorescently labeled protein, such as, again, BSA, is not enough for the uptake. These coacervates don't allow the entrance of this BSA into the job. So to wrap up what I have been talking about, this concept of biopizza produces cell-like structures using an enzymatic atom transfer radical polymerization. We can encapsulate macromolecules, particles, nano, micro, even bacteria. Most importantly, we can encapsulate a cell lysate that is functional in the sense that it can still have a metabolism to produce proteins. We are achieving or controlling the size and the shape of these vesicles. We can achieve a hierarchical organization, so compartments, new membranes, and whatnot. And future work will be on the growth and the division of these artificial cells to bring the concept of self-sustaining artificial cells to the polymer world. For this, I am finally ending. I would like to acknowledge and thank my former supervisor, Professor Bruns, my very good collaborators, Dr. Gimaja, my students, Christopher, Dominic, and Andrea, our collaborators from the University of Basel, those from Mainz, the Bruns group here in Darmstadt, and all the funding. And I am done with presentation. Thank you very much for your attention.
Thank you. Thank you so much. That was awesome. There are questions in chat. Can you see chat? If you could read, if you can pull it up and read a question out loud before you answer. Oh, and you have a question live too. We can start with that.
Okay, sure, please.
If you want.
Maybe the one live, and then I go to the.
Hi.
Yep. Hi.
Hi, I'm Ketan. Yeah, excellent talk. So I have a question regarding the actin expression in the vesicle. So can we actually couple the myosin production in the vesicle itself to create the ectomyosin machine, to create the stress fibers?
your audio was cut out. I didn't understand a few words there.
Okay. Okay. Is it audible now?
Try to talk.
You're breaking up a little bit.
Yeah.
Oh, okay. So can we actually couple the myosin production as well in the vesicle with the actin?
I mean,
to
would be
create
lovely.
the stress fiber inside the vesicle so that we can actually control remotely.
Yes. So first of all, I must say that this was a very crude polymerization that we tried. We didn't even use ATP itself to do the polymerization of actin. We used cations, so the very basic one. Ideally, yes, it would be extremely nice to produce more than one cytoskeletal protein, like myosin, to have something that actually allows the movement. Then, of course, with the movement, you have to think about how do we induce some polarity, or how do we break the symmetry of a vesicle. This, however, will take many years, and hopefully I will have the time to work on that in my career. But yes, that's something
that will be interesting. Yeah.
So let me just move to the yes,
so in the M clover production, so you have shown the graph for the fresh GUVs and
I'm very sorry. Your voice keeps breaking.
Their activity.
Would you mind writing the question in chat? I think it would be easier.
Have you tested the activity of that enzyme?
Sorry, Ketan, I don't think the GUV we can hear you. No.
Just to compare.
I think you might need to write your question in chat.
I think it would be easier. I'm sorry. Write your question and I will get to you immediately. So let me go to the first question by Eva. First of all, thank you. In that experiment, did you see the dependence of actin polymerization behavior on the compartment size? I did not. I must say that I didn't really check it too deeply. We did see that smaller vesicles, for M. clover at least, tend to produce a bit more protein, judging at the fluorescence. For actin, I didn't have the tools at disposition to quantify it per vesicle. But yes, that's something, again, the size dependence on expression that would need to be investigated. But also, that's also why I'm trying to make bigger and smaller vesicles in order to keep something a bit more homogeneous. And then from Angelica, what is the pH dependency of the creatine kinase reaction? How does that intersect with the requirement for cryosilvation? Oh, okay. So this, I must admit, it's something that my collaborators would know better. We do see that by changing the pH using another reaction, that is the urease versus glucose oxidase reactions, where we can increase the pH and lower the pH. Yes, we are able in that case to dissolve the cryosilvate or induce the cryosilvation back again to an extent, not perfect yet. But still, for the creatine kinase, we were working at neutral pH for the moment. But yes, good point, good question. I don't know if different pHs would change this reaction.
I think that's all the questions you had. I don't see any other. I just want to say thank you so much. That was really inspiring talk for me, because shows us kind of the boundaries of engineering of the encapsulation systems, and not what everyone just thinks lipids. And
thank you for your invitation. It was a pleasure to talk about my work, and I'm always happy to talk about future development of this research, which is very much in its infancy. So please be kind if you see some things that are maybe not perfected or very rough. If they are, it is. And I really hope that I will have the chance of improving everything and investigating even more questions about this.
it's awesome. Thank you so much, and good luck developing that system.
Thank you. Have a nice day, and thank you everybody for joining.
Have a nice day, everyone. Bye.
Bye.